After studying this module, you will be able to know about the modes of collection and preservation of drug evidence, learn the importance of packing of evidences, and identify the correct mode of collection and packaging of the drug evidence. First, the introduction. Present analytical methods can provide the forensic toxicologist the capability to respond to the queries that earlier were considered either despairing or not worth considering for the reason that the consequences were so time and again negative. Approaches that are sensitive to nanogram extents of drugs make it meaningful to carry out an analysis, even when the plate, cup or container involved has apparently no food or drink left in it. Drugs may be detected in blood at therapeutic concentrations, so it is imaginable to obtain indications to the medical history of the deceased, the victim or the suspect, even when they are not capable or reluctant to provide this information for themselves. Therefore, the detection of drugs used in the treatment of epilepsy, diabetes, etc. in a blood sample taken from an unidentified body may initiate a fresh sequence of explorations that leads to efficacious identification of the body. In the same way, allegations of intoxication prior to rape or burglary may be established. Since drug specimens display a wide multiplicity of detectable features, they may substantiate other evidence or link a suspect with crime scene. While handling this evidence, it is essential to avoid any contamination among the specimens or from other sources. The Forensic Science Laboratory handles the examination of marijuana and other drugs which may be involved in criminal cases or found in the custody of subject involved in a number of crimes. If there is abuse of drug by medicinal preparation and it was found in prescription boxes or bottles, it should be left in their containers which can be sealed and marked individually. It is imperative that every single item submitted as evidence must be sealed with tamper-proof evidence tape, dated, initialized and labeled as to its content and its association with the victim or suspect. If any evidence is stained with body fluids such as blood, semen, etc., it must be air-dried entirely prior to packaging and submission to laboratory. All items should be separately packaged and sealed in breathable, non-plastic example paper, loose-fitting containers. Estimated weights of drugs should not be documented on the request for examination of evidence form. Next is forensic significance. Maximum cases that go in a forensic toxicology laboratory start with the suspicion that a drug or poison is present. A casualty might be an accident, suicide or murder, but a toxicological examination must be carried out to support the investigating officer to resolve which of those it might be. Often the investigating officer will not know whether or not any offense has been committed until the results of the toxicological analysis are available, so that formulating the correct questions to ask is vital if accurate and useful answers are to be given. Therefore, particulars of the circumstances that lead to the conclusion that a criminal action might have taken place must be supplied to the toxicologist so that the analysis can be scheduled. Samples need to be taken and packaged properly and handed to the toxicologist with appropriate storage through this manner. Failure to manage this stage properly can ruin any subsequent analysis. The pathologist carrying out a post-mortem examination will be knowledgeable in knowing which sample should be taken for toxicological analysis. But cautiousness should again be applied to make sure that samples are placed in suitable containers and stored correctly before and during transportation to the forensic toxicology laboratory. It is essential that the correct samples be collected as soon as possible, properly and informatively labeled and stored correctly. Their procurement, storage and transportation to the laboratory should be documented adequately to guarantee a safe chain of custody. The containers used for the samples may be different depending on the analysis to be performed, and it is vital that the correct types are used. For example, a fluoride oxalate sample tube for blood is useless if fluoride estimation is required, and gamma hydroxybutyrate may be destroyed by citrate. If containers are to be inspected for fingerprints and for DNA, this should be carried out before any toxicological examination. The proper collection, packaging and transporting of urine specimens is a crucial part of a drug testing program. Although this is the most often challenged aspect of the process in any legal proceeding, it is also the least controlled one. Next important aspect is sampling of biological evidences. Appropriate collection, sampling and accurate storage of biological evidence are important, yet occasionally ignored. Steps in forensic toxicology. These factors, in combination with drug stability, can strongly impact the interpretation of results and the conclusion of forensic casework. Selection of the appropriate specimen is a critical component of any toxicological examination. Circumstances surrounding the case, the availability of specimens, the nature of the investigation, and even legal or statutory issues may dictate the selection and purpose of the specimens. It is essential that the specimen container is appropriate for the anticipated use and does not compromise the analytical findings. Container size should be appropriate for the volume or weight of the specimen so that the headspace is minimized. First biological evidence is blood. Blood is one of the most important specimens of toxicological concern, as it provides exclusive advantages over other matrices in terms of the extensive range of analytical methodologies available, the vast amount of available reference data for both antemortem and post-mortem drug concentrations, and the imperative value of the metrics from a pharmacological perspective. However, antemortem and post-mortem blood samples are remarkably dissimilar, and the site of the post-mortem blood draw, it can be central or peripheral, can be of critical significance. Femoral blood is the best specimen for use in post-mortem testing and should be sampled wherever possible. Next biological evidence is bile. Bile is by and large extracted from the gallbladder using a hypodermic syringe. It may be compulsory to tie off the gallbladder prior to collection if contamination appears to be a problem. Bile should be collected prior to the liver specimen to avoid contamination. Many drugs of forensic interest accumulate in the bile, particularly those that are heavily conjugated, such as opiates, benzodiazepines and cannabinoids. Bile may also be used in cases where chronic heavy metal poisoning is involved. However, owing to the presence of bile salts and fats, drug extraction from this matrix can be complex, and extensive extraction and cleanup procedures are often required. Next biological evidence is urine. In antemortem situations, a midstream urine sample is customarily collected into a plastic container containing sodium fluoride as preservative. In some situations, it may be essential to take safeguards against specimen contamination. In post-mortem situations, urine is collected by insertion using a hypodermic syringe directly into the bladder under visualization. puncture of the abdominal wall should be avoided to reduce the chance of contamination. Urine is valuable specimen for both antemortem and postmortem drug testing because it is a comparatively simple matrix. However, the diversity of factors influencing urine drug concentrations, example urine volume, clearance, metabolism, pH and time of last emptiness generally means that in isolation these results have limited quantitative value. Next biological evidence is kidney. Most drugs pass through the kidney as a result of urinary elimination. Kidney is an important specimen in cases of alleged heavy metal poisoning owing to accumulation in this tissue. Next biological evidence is liver. Liver is a particularly important organ because of the very large number of drugs that go through hepatic metabolism and the equally extensive available reference data that exist. To reduce the chance of drug diffusion from the small bowel, tissue from deep within the right lobe is chosen. The concentrations of drugs and metabolites in liver are often raised. Therefore, this specimen has limited interpretive value. However, liver is mostly useful for highly protein-bound drugs, and the evaluation of liver or blood drug ratios may allow the differentiation of acute overdose from chronic drug use for some drugs. Next is gastric matters. Gastric matters are a potentially valuable specimen for examination in postmortem and medical cases. Unabsorbed drug or tablet fragments in the gastric contents may provide valuable information relating to ingested compounds and provide an excellent material for preliminary screening owing to the potentially large amount of drug that may be present. The non-existence of a drug in the gastric contents does not necessarily exclude oral administration. The presence of a drug in gastric contents, particularly at low concentration, does not necessarily indicate oral administration. Drugs may be absorbed into the stomach by gastric juices that are in equilibrium with blood or as a result of intranasal drug use. Basic drugs are more vulnerable to this because they have an affinity to become trapped in the gastric compartment owing to the low pH. Next important biological evidence is muscle. Muscle is not normally came across regardless of the fact that it often contains fairly high drug concentrations, mainly for substances with high volumes of distribution. Perfusion rates between sites and drug concentrations are not consistent, and drug concentrations must be interpreted consequently. Muscle is encountered more frequently for ethanol determination in the absence of blood or in the course of the investigation of a suspected injection site. Next biological evidence is hair. Hair has been used in a variety of antemortem toxicology situations to provide a history of drug exposure and has therefore found applications in workplace drug testing, in observing of individuals on probation or on conditional release for drug use, in insurance testing to verify the reliability of statements made by applicants relating to whether they use drugs or are smokers, in child endangerment, in re-granting of driving licenses after suspension for drug misuse, in drug facilitated sexual assault and in other types of criminal casework. Here may allow drug exposure over several weeks or months to be determined depending on the length of the hair. Although head hair is the preferred specimen, hair from other sides, example pubis and axillae, may also be used. Next aspect is preservation of evidences. Specimens should be stored at suitable temperatures with suitable preservative. and in an environment accessible only to authorized person to make sure of the security and integrity. Short-term storage at refrigerated temperature, that is 4 degrees Celsius, is suggested for most of the samples, or frozen at minus 20 degrees Celsius, or frozen at minus 20 degrees Celsius or lower during long-term storage, that is more than 2 weeks. Exceptions to this contain hair, nails or dried blood samples on filter paper, which can be stored at ambient temperatures, whereas medical specimens are characteristically unpreserved. The use of a chemical preservative is often acceptable in forensic specimens. Preservation of blood samples with sodium fluoride is routine in most laboratories. Commercial evacuated blood collection tubes contain sodium fluoride as the preservative and potassium oxalate as the anticoagulant. These are the chosen evacuated blood tubes for ante-mortem forensic toxicology casework. Inhibition of microorganisms and enzymes with sodium fluoride is important for frequently encountered analytes such as ethanol, cocaine and others. Fluoride acts as an enzyme inhibitor and helps prevent glycolysis. All samples should be appropriately marked for identification with the case number, donor number, date and time of collection, signature or initials of the collector and specimen description. Tamper-proof containers or tape bearing the collector's initials and date of collection should be used. Specimens should be forwarded to the laboratory in appropriate leak-proof and tamper-proof packaging or shipping materials with all appropriate documentation like chain of custody forms, requisitions for testing, special requests, case information, medications list, police report, donor information, identifiers such as date of birth or social security number, agency case number, pathologist or police officer name and contact information. Improperly packaged or identified materials should be returned to the submitting agency. Documentation accompanying the specimens should list all of the specimens that were collected or available for testing. Once received by the laboratory, the specimens should be examined and appropriately documented in terms of condition and quantity during the assessing process. We will conclude this module with summary. Drugs may be detected in blood at therapeutic concentrations, so it is possible to obtain clues to the clinical history of the deceased, the victim or the accused. Drug specimens exhibit a wide variety of identifiable characteristics which may be corroborated with other evidence or link a suspect with the crime. If there is abuse of drug by medicinal preparation and it was found in prescription boxes or bottles, it should be left in their containers which can be sealed and marked separately. It is important that each item submitted as evidence be sealed with tamper-proof evidence tape, dated, initialed and labeled as to its content and its association with the victim or the suspect. It is essential that the appropriate samples be collected as soon as possible, correctly and informatively labeled and stored appropriately. Their acquisition, storage and transportation to the laboratory should be documented adequately to ensure a safe chain of custody. Specimens should be stored at appropriate temperatures with adequate preservative and in an environment accessible only to authorized personnel to ensure security and integrity. All samples should be properly marked for identification with the case number, donor name, date and time of collection, signature or initials of the collector and specimen description. Documentation accompanying the specimens should list all the specimens that were collected or available for testing.